Hello everyone, this video will be about chapter 10.2 of your book 2B. So it will mainly focus on the sense organ, eye, and the respective sense produced called sight. So here in this section we'll discuss how sight is produced and what affects our ability to see different things, alright? So first we know to see things we need our eyes. So first we have to learn the structure of the eyes. So it would be a bit difficult because a lot of new terms for you, so a lot of new words, but please remember so how to spell or which word correspond to which structure, alright? So first, when you only see the outer surface, alright? So you can't see the inside, alright? For the bottom figure, it is the inside, so you can't basically see that many structures. So for on your face, alright, on your face you have eyebrows. So it's the hair above your eye, alright, eyebrows. And then eyelashes will be the hair that grows on your eyelid, and then slightly you can touch them near your eyes, alright, and the eyelid, alright. So these are the three structures you can see on your face. So the eyebrows mainly prevents sweat, alright, prevents sweat from running into your eyes. For the eyelashes, it will prevent some small objects or like sand or dust from entering the eye. And for the eyelid can control opening or closing, right? You can close your eyes with the eyelid and to protect the eye from dirt and also strong light. So you can see the structures on your face, a lot of them is used to protect your eyes, alright? So remember eyebrows, eyelashes, and eyelid. Okay, so after talking about the surface, so the eyebrow, eyelid, and eyelashes, let's get into the eyeball. So where are the eyeballs located? So each eyeball here is held in the socket of the skull called the orbit. So where exactly is the orbit? So here you can see the skull, right? You often see like maybe pictures or even real samples of the skull. So the two holes, big holes here, exactly are where the eyeballs are placed, alright? So these two holes will be called the skull. And your figure 10.3 will be the side view, alright? So you cut from the middle and then look at from the side, alright? So it will be like this, alright? Your figure 10.3. So you can see the eyeball placed in here. The whole space is called orbit, and then there are three pairs of eye muscles, alright? Remember, three pairs of eye muscles. So the muscles can allow the eyeball to rotate in different directions. So you can move up, move down, move left, move right, alright? So these are the outer structure of the eyeball. And next we'll talk about the structure, the details of the eyeball. So we focus on this thing, alright? So now we are talking about the whole outer structure, how it is located and how it is protected. So next, the experiment 10.1, alright? Practical 10.1 will tell you the details of the eyeball. So you can see the eyeball here. So this is the one that is placed in the orbit. Okay, so and you can see question 2 here. Label the diagram below using the words provided. 9 new words, alright? So to fill in these boxes. So please remember these new terms because each word represents each part and each part has a unique function, alright? So A here, cornea. So we refer to the outermost white layer here. And B, iris. Iris will refer to the two structure here, alright? This one, this colored structure is called iris. And pupil. Pupil only refers to the hole, alright? The opening here. So this opening is called the pupil. So we'll go through how this pupil and iris works, alright? In the later part. And D, lens. So this white thing is called a lens. And then jelly-like fluid. So the whole eyeball, because it is not collapsed, right? It has some space in here, and the space is filled with something called the jelly-like fluid. And retina. Retina will be this here green colored area here. And sclera. Sclera will be the outermost white layer, but not in the front. Alright, in the front it is called cornea, and then on the side it will be called sclera. And H is called blind spot. So blind spot is a place where the optic nerve, so eye, you can see the optic nerve here, leaves the eyeball. So this is called a blind spot. So now you learn how to name different structures of the eyeball. So now you have learned the names of different parts of the eye. So the next part would be the functions, alright? So what are they used for? So we'll go from the outside to the inside. So first, outside, remember the front part we call cornea. So the cornea is a transparent, so allows the light to enter the eye so that the eye can function. And also, there's a little bit curved surface you can see here. So it's not straight like this, so it's curved. So it can help focus the light. And onto the back part, alright, it's called the sclera. So sclera is just a tough white coat. So it's just like a cover. So it protects the eyeball. So this is the outermost part, alright, so this white layer here. Okay? And then moving in, we have the structure called the iris and the pupil, alright, iris and pupil. So iris, it controls the size of the pupil. So these two are related, alright, so the iris is used to control the size of the pupil, and pupil, what's the use of pupil? It's just an opening at the center of the iris, so to allow the light to pass through the eye. So the pupil has another function, or the iris and pupil together with other function is in here, alright, so I'll talk in a minute. So let's focus on other parts first. So we have learned cornea, sclera, iris, pupil, and then lens. So the lens is just like your glasses. If you wear a pair of glasses, it just focuses the light, okay? Focuses the light onto the retina. The retina will be this layer, alright? So we'll go through that in a minute. And jelly-like fluid just maintains the shape, so to fill the eyeball, alright, with a fluid, alright. And the retina. So retina will contain something called the light-sensitive cells. So light-sensitive cells are exactly those specialized sensory cells we have talked just in the last video, right, in chapter 10.1. So these cells are necessary to produce the sight sensation, or the sensation we call sight, so they can detect different light. For retina, it contains light-sensitive cells, yes, but then there is a point that no light-sensitive cells are found there on the retina. So this is exactly the point we call the blind spot. So because at this point the optic nerve will leave the eye, so there has to be an opening, so there will be no light-sensitive cells here. So when light shines on this part, there will not be any message sent to the brain. And the optic nerve, why we need that? Because the message from the light-sensitive cells are sent along this structure to the brain. So when messages are sent to the brain, the sensation called sight will be produced. So these are the functions of these 9 different parts of your eyes, alright? So for the pupil and iris part, so here you can see the bridge to the DSC biology. So it explains more about the relationship between iris and pupil. So basically, iris controls the size of the pupil, and in different environment, the size of the pupil will change in order to let more light or less light to go into the eye. So in dim light, that means a darker environment. So imagine darker. If you want to see things more clearly, then you have to allow more light to enter your eye so that the cells there can receive the stimulus, right? So in order to let more light to pass through or enter your eye, so your pupil must enlarge, right? So the pupil must become larger in order for more light to pass through and enter the eye through the lens. So in dim light, the pupils will become larger. And in bright light, or on the other hand, then you want less light to enter your eye, or else too much light going in, it will hurt your eye, right? So the pupil will become smaller and prevents the eye from being damaged by a strong light. So this is more of the functions of the pupil and the iris. In practical 10.2, it concerns about the dissection of an ox eye. So it sounds a little bit gross, but then an ox eye, the structures of ox eye and human eyes are very similar. So through the dissection, you can know more clearly about how each structure is placed inside the human eye. So it's not going to be done in school laboratory, but you can watch the video. So it clearly tells you how to dissect and cut open the ox eye, and visualize the things inside. So you pay attention to the video and I'll go through the answers with you right away.
This is an ox eye. Look at the eyelids, the eyelash, the eyeball, and the cornea. Use a pair of scissors to cut away the eyelid, the fatty substance around the eyeball, and the eye muscles. Cut through the cornea diagonally. Display the cornea. What is the color of it? Then cut through the iris and the sclera. Take out the lens. What is the color of the lens? Put the lens on a piece of newspaper. Look at some print through the lens. Does the print change in size? Take away the jelly-like fluid to expose the retina. The part on the retina where the optic nerve leaves the eyeball is the blind spot.
So you have watched the video how he cuts open the ox eye, and you can also see every structure he's showing to you. So the first one about the sclera. So press the sclera gently, so you cannot press, but you can see that when he cuts open the eye, the sclera is actually very tough, so it will not collapse or change shape. So it can maintain the shape of the eyeball. So actually, yes, it is tough. And on to the next page, it will be about the front half of the eye. So identify the cornea, iris, and pupil. So cornea is exactly the one that is cutting across, so diagonal cut, and then he flips open 4 pieces of colorless thing. So that colorless transparent thing is the cornea. So for the cornea, the color should be colorless, alright? And the iris refers to the black substance like around or inside found in the eyeball, right? So the black thing is actually the iris. So color of iris should be black or dark brown depending on your sample. Usually for ox eye should be black. So you can see in the video that there's an eyeball filled with a black thing. The whole black thing is the iris. And then the lens, you can see it takes out again colorless transparent thing. So that is the lens. So the lens should be colorless again. And then you put the lens on the newspaper and look at some of the words. So on the right you have the picture here. So you can see, compare the eyes, so these letters with the remaining letters, these letters are much more bigger, right? You can see more clearly. So they becomes bigger. So the word changes in size and it becomes bigger. So it is like a magnifying lens, alright? So to magnify some small objects or small words. So for the lens, it is not a very tough structure. So if you press the lens gently, so the lens actually will change, the size would decrease. So you can change the size of the lens by giving it some force. So now you know more clearly about the lens, the cornea, and the iris. So the lens is the most important thing you have to know, because by controlling the size of the lens, it can help you focus on different objects or look at different objects clearly. So about the function of the lens, in the later pages you will know more about how lens actually works. And for the remaining structures, including the retina, optic nerve, and the blind spot, it is at the back of the eyeball, basically used for producing or receiving the stimulus and then producing the sensation, called sight. So this basically wraps up for practical 10.2. So you have learned from the previous practical each structure of the eye. And the next most important question is how it works, so that we can see different things. So now let's look at the animation first. It will talk about how light enters the eye and how it is focused onto the retina to produce vision, or what we call sight.
Our eyes are the sense organs that detect light. They are the most important sense organs in our body. Now let's see how our eyes work. The doll reflects light, and the light enters our eyes. The cornea, the lens, and the jelly-like fluid bend the light to help focus the light onto the retina. A sharp and inverted image is formed on the retina. On the retina there are two types of light-sensitive cells, namely rod cells and cone cells. Once these cells are stimulated by light, they send out messages. The messages are then sent along the optic nerve to the brain. The brain then interprets the messages as an upright image of the object.
Okay, so you have watched the animation. So let's talk about the 4 steps, how sight or vision is produced. So of course, first light has to enter the eye. So light enters the eye. Remember, when there is no light, you can't see anything, right? So it is all dark, you can't see anything. So because light from the object enters our eye, so we can see things. And then the second process is where the lens plays magic, alright? So the magical lens, how it can helps produce the vision or the sight we want to see. So the lens actually can bend the light, alright? Bend the light. So when the light, you can see, travels from here, it suddenly changes direction and goes to here, alright? So following the blue lines. So this is how lens works, alright? It can bend the light and focus it onto the retina. And in the process, remember, an inverted image is formed. Inverted. So you can see this arrow, it is upside down, so the top part becomes at the bottom, and also left right is inverted. So the red part is originally on the right and now it's on the left. So this is called an inverted image. Okay, when the inverted image is formed on the retina, then because there are light here, right? Remember when we talked about the sensory cells? So the sensory cells here, we have a name called the light sensitive cells. So as suggested by the name, is called light sensitive. So they should be stimulated by light, as you have learned. So they will send message to the brain along the optic nerve. So when an object is formed here, because there is light, so the retina, the cells on the retina called light sensitive cells will be activated. So they are told, you start working, and then they will send the message to the brain through the optic nerve. And then the brain can interpret the message as an upright image. So because remember, it is inverted, right? So when it travels to the brain will know, oh, I have to invert it again, so it becomes upright. So remember, the opposite for inverted will be upright. So basically, the yellow box will conclude the process how we can see, alright? So first light from the objects, and then cornea and the lens will be the one bending the light and focus onto the retina. So remember, bend the light. And then the retina, it contains a cell, the sensory cells we call light sensitive cells. And then they will be stimulated by the light and send the message to the brain to interpret the message as an upright object. So remember, inverted and then upright again. Just now you have seen that lens can change its shape, right? And it is exactly because of this property that we can see both near, that means close to us, object close to us, we call near objects, and also distant objects, that means objects that are far away. So by changing the shape of the lens, it can actually change the way it bends the light, because when light passes through the lens, it is being bent and it changes direction. So how it works, it's actually a very difficult physics principle, alright? So it involves how the light passes through something and changes direction. And this extension is actually about this part. So I'm not going through every single detail. I'm showing you the video so you can see how light changes direction when it passes through lens of different shape and also different thickness, alright? And I'll go through the answers with you after you've watched the video. So you have watched the video and I'm going through the first two answers first. So the distance between the light source and the lens, so it's the thin lens, should be 20 cm. And between the light source and the thick lens should be 10 cm. So for this part, it is this 20 and 10 is actually because of the focal length of the lens, but this is a very advanced physics concept, so I'm not going through this here. But what you need to know is that thick, thin lens, they got a different proper distance for focusing objects. So you can think in the other way around. So if you have objects that are nearer, then from this experiment you should use a thicker lens. So the thicker lens should have a shorter distance, right? Remember, the thick lens only 10 cm, thin lens you got 20 cm. So if the object is only 10 cm, you use a thick lens. This is exactly how this practical works. So if the objects are further away, that means distant object, you should use a thinner lens for focusing. So this is how lens can change its shape, or in more accurate terms, you should change the thickness, alright, for focusing different objects. So, and this picture just concludes what is just mentioned. So when seeing near objects, the light from a point of a near object, so the lens will become thicker and then it directs onto the retina. And light from a point of a distant object, it will be like a parallel line, so you need the thinner lens. So how to draw these diagrams, I'll not test you on this, but remember the lens is elastic, alright? The lens is elastic. So it's mentioned here, lens is elastic. So it can change the thickness, so that when you see near object, you use a thick lens. When you see distant object, you use a thin lens. So this is exactly what this extension wants to tell you. Okay, in the yellow box, just a quick conclusion. So remember, one very simple thing: near objects, thicker lens; distant object, thinner lens. That's the whole concept. And for the next part will be about seeing colors. So just a minute ago, then you have watched the animation and it talks about the concept that when light reaches the retina, there are two types of cells called the rod cells and the cone cells. So actually, when we see objects, we are able to see different colors, right? Red, yellow, orange. So it is because we have cells that can identify colors, but we also have cells that cannot identify colors. So rods are the ones that cannot tell the colors. So they are only for black and white. And cones, they are able to tell the colors, alright? So they can tell red light, green light, and then blue light. And by mixing different combinations of red and green, you can also create other colors, just like what you have learned in arts, alright? And rod cells and cone cells, because rod cells only need black and white, so they can work in dim light, alright? And for cone cells, because they have to tell different colors clearly, so they have to work in bright light. So these are the brief explanation of the two different types of light sensitive cells on the retina. So remember, rod cells for black and white, cone cells for color. And rod cells are sensitive to dim light, so no need a lot of light. But then cone cells sensitive to bright light. So this extension gives you more information about how we can see different objects, alright? And on to the next page will be a little interesting fact about bullfighting. So someone waving a red cloth in front of the bull and then it stimulates him. So actually for bull, it cannot react to the red color. So no matter what color is the cloth, if you wave it like that, the bull still comes to you. But then why it is red? Because a red cloth is more attractive. So this is the fact. So just an interesting piece of information for you to know. And for the bottom part will be checkpoint 2. So pause the video now and try to do the matching and also the true or false question. I'll go through the answers with you right away. Okay, so just quickly go through the answers for checkpoint 2. So the first one, matching, match the structures with the description. So for cornea should be the one that is a transparent layer to help focus the light. So it did a little bit help in focusing the light, but not the major one that helps bend the light, alright? And the second one, iris. So iris, remember, it controls the size of the pupil, just introduced in the previous sections. And for pupil will control the light, amount of light entering the eye. So it is an opening that allows the light to enter. So it should be number 6. And lens is the one that can bend the light, remember? So the thickness can be changed to focus light onto the retina. So it should be the third one, number 3. And for the retina, it will be the one that contains the light sensitive cells, remember? So the retina should be number 2, contains the light sensitive cells and to receive the stimulus. And optic nerve will be the road that the message is sent through, alright, through the optic nerve to the brain. So message from the light sensitive cells are sent along this structure to the brain. So F should be number 4. Okay, write true or false for the second question. So image form on our retina are upright, so it is false. Remember, image on the retina is inverted, and then when it is inverted, it's sent to the brain. The brain can know, oh, I should invert it again, so it becomes upright. Remember, inverted, not upright. There are 3 types of rod cells on our retina, so it is also false. Only one type of rod cells, alright? So this basically concludes checkpoint 2. I've just now talked about how we can see things, but then of course we cannot see everything in this world at the moment, right? So we have limitations. So there are actually a lot of limitations of our eyes. So for activity 10.12, it mentions how close and how far you can see. So that means the distance, alright? But actually, apart from the distance, there are also other limitations. So actually there are 3, we can say. So I'll talk about these 3 now. Okay, so written in your books, there are different situations where we cannot see things clearly. So the first one is about too near, second one too far away. So these are all about the distance of the object we want to see. So apart from distance, we know that if the objects are too small, then we also cannot see them clearly. So we know that there are at least 3 limitations now you learn. So too near, too far away, or too small. So for too near, it is easy to solve, right? We just pick up the object and then place them further away. So the distance is proper for us to see it clearly. But then for too far away, we cannot reach the object, so we cannot make them closer. And also for objects that are too small, we cannot make them bigger randomly, right? It's difficult for us to make things bigger. So in order to solve these problems, we need different instruments, alright? So there written in your book, there will be some examples. So for hand lenses and microscope will be used to solve problems that are concerning about small objects, alright? So help us see small objects. For hand lenses, the ability to magnify will be lower, but for microscope, it will be much more useful in magnifying really small objects. So hand lenses can be the ones used by your grandpa to watch newspaper, but for microscope, usually used by scientists to see like cells. So really small, you cannot see with bare eyes. Okay, and concerning about far away objects, so we use binoculars and telescopes. So for binoculars, it is usually used to see things like 1 or 2 or hundreds of km away, so not really that far away, but still a very great distance between you and the object. But then for telescope, it is usually used to see stars or planets in the universe, so it will be thousands or millions of kilometers away. So the applications are different. So for binoculars, used to see bird flying, but telescope like used to see details of a planet in the space. So these are all used to help us see far away objects, but the applications are just different. So remember the examples, and in the yellow box there will be a short conclusion. So we cannot see clearly when an object is too near, far, or small. Alright, 3 situations. And use different instruments. So remember the 4 examples to extend our vision. And so on this page on the top, it is just some history about the development of microscopes. So from very brief ancient type microscope to the modern microscope you see in the like in many universities or laboratories. So the magnification power is also different. So just if you have interest, you can read this little box. Alright. So the next topic about vision is about sight is called blind spot. Alright. So apart from the limitations, there is another limitations. Alright, not about the object but about our structure. Alright, it's called blind spot. Alright. So what is blind spot? In activity 10.3, you can try to follow the steps, and you will find a very interesting phenomenon you can see. So actually, if you keep looking at the frog and then move the book towards your eyes slowly, then there is a point where the mosquito will disappear and then it reappears again. So this is exactly what will happen if you can try to follow the route, the instructions, and you will see what exactly would happen. So why? It is because of the presence of the blind spot. Why the mosquito would disappear and then reappear again suddenly? So it is because at a certain distance, the image, or we say the light, of the mosquito will fall on the blind spot. So what exactly is the blind spot? Actually, the blind spot is the point where the optic nerve leaves the eye. So remember, we have an optic nerve here, so the nerve is responsible for sending the signals to the brain, but then it needs a place to connect to the retina, right? So there must be a point where it leaves the eyeball and then goes to the brain, and at this point there are actually low light sensitive cells. Alright, so you know light sensitive cells are used to detect the light, right? So if you don't have these cells here, it cannot detect the light. So in case when the light pass through the lens and then change direction and falls onto this point, then because there are no cells, you cannot detect anything. So the eye will think that, oh, there are low lights, but actually there is, but just you cannot detect them. So because low light sensitive cells there, so no stimulus detected, and so no message will be sent to the brain, so you cannot know there are objects there. But then it is there, but just because you don't have cells here, so no message is sent. Alright, remember, at a blind spot, even if there is light, no message is sent to the brain when an image is formed on it, so we cannot see the object. So this is exactly why at some point we cannot see the object. It is a very specific point. Alright, not very big, but for the blind spot it is how it works. Alright, so we cannot see objects if the image is formed on the blind spot. And for the misconception part, it's just to help you understand how to write something. So you cannot say that no images are formed, because actually the light passes through your lens and falls on the blind spot. So it works the same way as it falls on different places of the retina. So actually there is light and actually there is image formed. But then because there are no cells, remember, I say it again, there are no cells, alright, no light sensitive cells, so you cannot detect anything. So this is how blind spot affects our vision. Okay, and about the extension part, so this part I will go through a little bit of them. So it's about defects of the eye. So we know a lot of eye problems, right? So that's why a lot of you wear glasses. So look at your class, look at your classmates, a lot of them are wearing glasses. So it is because of defects of the eye. So I also wear glasses. So why there are defects of the eyes is usually because of the problems in the lens. So first I'll cover the first one, is called short sight and long sight. Alright, So short sight, basically, if you have short sight, you can only see close things, alright, near objects, but you cannot see distant objects. This is very common. And the opposite will be long sight. You can only see far away objects, distant objects, but not near objects. So this is actually because of some problems in the lens, alright. So in practical 10.4, it is an experiment to demonstrate short sight, long sight, and the correction method. It is actually a very complicated experiment, so please pay attention to the video to make sure you know what it is doing, alright? And you can replay the video more times if you don't know what it is doing. I'll give you a really short version of explanation about what actually it does to demonstrate short sight and long sight, and what causes short sight, long sight, and how to correct them. So please pay attention to the video first. Okay, so you have watched the video and you can see a similar device or setup in the video, right? So there are three lenses: lens A, so normal eye; lens B, short sight; lens C, long sight. So lens B should be thicker than lens A, so you know that short sight is related to a thicker lens, alright? If your lens is thicker, that means short sight. So lens C, long sight, so should be the opposite, thinner. So thinner lens, long sight; thicker lens, short sight. Okay, so remember this. Okay, so for normal sight first, so turn on the light source and then watch into the glass through lens A. So lens A means normal sight. So if you don't need to wear glasses, that means you have a good eyesight, normal eyesight. So which part of the eye does the back of the flask represent? Of course, it should be the retina, because the light would converge or change direction and then stimulates the cell on the retina, right? So the image should be formed on the back. So this back will represent the retina. And on to the next page. So about short sight and long sight. So the diagram should be like this, alright? So for a thicker lens, the light is focused in front of the back of the flask. So the image form, of course, is not sharp, right? So this point is sharp. So the light and then diverge, okay? Diverge again. So diverge means they go to different directions, alright? So it is not a sharp image form. And you place a concave or a convex lens in front of lens B, alright? So which lens? So from the video, you should know that the concave lens should be okay. So by completing the diagram, it would be like this, a little bit complicated. But for concave lens, so the function is to make the light goes to opposite direction, alright? So you can see here when the light is parallel and then it goes to opposite direction, alright? So they become separate more. And then the lens can change the direction again, so that after this correction, the sharp image will be formed at the back of the flask. So it is how concave lens work, remember, concave, alright, to solve the problem of short eyesight. That means a thicker lens like lens B. So make sure you understand this part first. So if you don't understand, go back and watch the video again. Okay, for lens C, it will be about long sight, right? Long sight. So you can see long sight actually at the back. So of course, because it is passing through the flask, so this point you cannot see, but at the back, again, the image is not sharp because light is focused behind the back of the flask. So it is not sharp. Only at this point it will be sharp. Other points are all not sharp, alright? And again, from the video you should see, put the concave or convex, should be a convex this time. So the diagram will be like this, alright? So the convex lens has a function of changing the direction, just acting like a normal lens, alright? So it can make the light goes stick closer. So you can see here it converge. Okay, it converge. So converge means they're getting closer. So it's getting closer first, and then the lens again change the direction again to make it get closer again. So it will help the light ray gets closer together in a much faster way, so it can again form a sharp image at the back of the flask. So this is how convex lens used to solve the problem of long sight. That means a thinner lens, all right, like lens C, all right. So remember two things, all right. So one is about short sight, one is about long sight. So short sight, thick lens, concave lens, 3 words, all right. Short sight, thicker lens, concave lens, yes. And then for long sight, thinner lens, convex lens. So this will be the whole idea about this experiment. Even if you don't understand how the light bends, but then you have to know lens and different eyesight. Okay, so here just some more detailed explanation about what short sight and long sight is. And you can see this diagram is exactly the same as the diagram you have seen about in the experiment part, alright? Just the flask and the lens is replaced with the eyeball. So if the lens is thicker, then it is in front of the retina. So thick lens, again, short sight is lens being too thick, alright? So you should be corrected by wearing concave lenses. And here it mentioned another problem is eyeball being too long. So eyeball being too long, again, the result would be image formed in front of retina. So remember, for short sight is image formed in front of retina. This is the most important point. So lens being too thick, eyeball being too long. So these two will cause image formed in front of retina. And so it's wearing concave lens, so to bend the light away, okay, so make them separate first and then converge, get together again. So this is short sight. For long sight, it is exactly all things being opposite, alright. So thick becomes thin, so lens becomes being too thin, or eyeball being too long, so opposite would be eyeball being too short. So all these point to the problem image formed behind the retina. So just like what happened to lens C, so the thin lens in that experiment. So image formed behind the retina, so behind the end of the back of the flask. So it is corrected by wearing convex lens. So it will make them go together, make the light ray go together first, and then again change the direction so they can get a sharp image formed on the retina. Okay, again in the yellow box, just a quick conclusion. So short-sighted problem would be near objects okay, but distant objects not okay. So because the lens too thick or eyeball too long, so you have to wear concave, so to make the light ray separate first, alright? So for long sight, the problem would be distant object okay, but near objects not okay. So exactly, remember, exactly the opposite of short sight. So lens being too thin or eyeball being too short, and you should wear a convex lens. So this is the part about short sight and long sight. And for the science file, it's just another technology to correct the problem about short sight and long sight. So if you have interest, you can read it. So it's exactly like a little surgery, so to change the thickness of your lens to help to correct the problem. So apart from short sight and long sight, there are actually other eye defects or disease. So the first one here shown is color blindness. So these disease cannot be cured that easily. Not wearing a pair of lens would help. So people with color blindness cannot distinguish some or all colors. Remember, some or all colors. The most common would be that they cannot distinguish between red and green. So this is actually a problem, or what we call an inherited defect. That means passing down from generation to generation. So maybe your father or your mother, maybe they get the disease and then they may pass it down to you. So this is what we call inherited defect. So remember, for some certain color blindness, not only like called red-green color blindness, so not only red and green will be affected, but other colors may also be affected or not so clear. And for activity 10.4, it will be some diagrams or photos used to test color blindness. So you can check to see if you can see the numbers here. So not all have numbers, so you can check if you can see it clearly. So this is a good test for color blindness. So the second one is called astigmatism. So astigmatism refers to blurred vision, alright? It is not related to distance. So no matter it is near object or distant object, they are all blurred for people with this disease. So it is usually because, again, of the problems of the lens. You just need to know there's such a disease. Okay, and the final disease to mention is called cataract. So cataract usually is when the lens becomes cloudy. So it will more likely to happen in elderlies, but not teenagers. All right. So cloud blindness, astigmatism, and cataract. So these three forms of disease. So you just need to know the names. All right, as an extra knowledge for you. Okay. So the last part of 10.2 will be about eye care. So about your sight will be how to take good care of your eyes. So in activity 10.5, just a comprehension, so you can read the paragraph and try to do the questions. I'm going through this very quickly. So one eye problem list here will be here, okay? So short sight might have a much higher risk of developing eye problems like cataract or retina damage. So cataract or retina damage. So causing short sight, another way of long-term use of electronic screen products may harm our health. So improper posture causing muscle pain, or loud noise may cause hearing problems. And one good way is keep enough distance between the eye and the screen, and also give the eye a rest, looking at distant object every 30 minutes. So these are just a very brief introduction about the problems related to eyes, and also how to keep your eyes in a good condition. So how to take good care of your eyes will be on the next page. So here are some tips. So just very quickly go through each of them so you can read yourself, and not something very special, so you may have heard of it or not. So here there are 6 points. So the first one, sufficient light. Of course, sufficient light is good for reading, and also keep enough distance between your eyes and any electronic screen product. So you're now using your phones very often, so make sure you keep a good distance between your eyes and the screen. And then again, give your eyes a rest every 30 minutes by looking at distant objects. And the fourth one will be about strong light. So if you have a very strong light, it may hurt your eyes, so wear sunglasses so when you're outdoors. And then do not rub your eyes because it can cause infection, and also have regular eye checks. So these are just very similar common actions you can take to protect your eyes. And onto the final page would be checkpoint 3. So now pause the video and try to do the questions. I'm going through the answers with you right away, so pause the video now and do it. Okay, so now I'm showing you the answers and going through each question one by one. So first part, true or false. So 1A, we can see small objects clearly with the help of a binoculars. So it should be false because it's not small objects. So remember, small objects using hand lenses or microscope. Distant object using binoculars or telescope. So binoculars should be distant objects. And 1B, there are no light sensitive cells on the blind spot. So it is true because on the blind spot is where the optic nerve leaves the eyeball, so there would be no light sensitive cells, so it is correct. C, images cannot be formed on the blind spot, so it is false. Again, we have talked about that. So image can be formed on the blind spot, but because of no cells, so there will be no message sent. Okay, for 1D, people with astigmatism cannot distinguish color, so it is false. So the disease should be called color blindness. So it's not astigmatism, it's color blindness. So for the second question, fill in the blanks. So about short sight and long sight. Alright, so remember, short sight is lens being too thick and then can only see near object but not distant object, and long sight would be exactly the opposite. So lens being too thick, 2A, should be cause of a short sight, and then eyeball being too short will be cause of a long sight. So remember, for short sight will be eyeball being too long, and the opposite for long sight, eyeball being too short. Okay, and for 2C, they cannot see distant object clearly, so it would be a problem for short sight. And by wearing convex lens would be long sight correction. Alright, correction of long sight will be wearing convex lens. So it is all about the section short sight and long sight. If you have any questions, just refer back to that part. Okay, and the last question, so it is a multiple choice. So it shows the path of the light from a distant object in a boy's eye. So this is exactly the experiment part, so it should be short sight. The answer should be A. So this concludes for section 10.2 about sight. So please make sure you understand everything in the video and finish the homework. Thank you.